It's a real pleasure to introduce this new ARF research project. Before I do, though, let me spend just a few minutes on giving you a little bit of context. I think a lot of people in this room, your ARF members, you know about our first Neuro project from 2011. It was the first project that independently looked at these new neuromarketing methods. It was a big success, I think, for the ARF. We got a lot of positive feedback, but that first study did leave two questions open. First, how do traditional methods compare to neuro? Do these neuromarketing methods add something to the traditional methods? That was not part of that first project. And the second question, which methods, traditional, neuro, or maybe a mix of those two, are best at predicting success in advertising? So this new project, which we call Neuro 2, is designed to answer these questions. And again, it's the first, we think it's the first, to the best of our knowledge, the first major study that looks at commercials tested in a variety of methodologies in a way that is comparable from the research protocol with the samples. So what you hear today is a study based on 37 commercials, and so far we looked at 48 different methods, measures, using all of these different methods. So more is to come, but we already got some really exciting data for you, and I can tell you, working on this project, which promises really to tell you advertisers and media companies, whether you do advertising or promotions, how to improve your methods for testing creative, to make them more effective in the marketplace. Really is an exciting project to work on. Of course, it's also very scary, because in a complex project like this, you never really know exactly how it's going to turn out, right? Well, you will see in a minute how it turned out, but before I get there, I do have to give a big thanks to all of our sponsors who went on this journey with us, who supported this project. A couple of you are in the room. I've seen a couple of you. So thank you guys for working with us and continuing to work with us on this project. So it's now my real pleasure to introduce from the teams that worked on this project, two scientists who are going to summarize it for you today in the time that we have. The first one, Vinod Venkatraman from the Temple University. He is the Associate Director at the Center for Neuromarketing at Temple. And Russell Weiner, who is the William Joyce Professor of Marketing, and he is in New York at the Stern School of Business. And Vinod, he's going to be the first one to summarize these results. And thank you, Vinod, and welcome, Vinod. And the stage is yours. Thank you.
Thank you, Horst. It's my pleasure to try and do the more complicated job of summarizing this complex study in the next 6 or 7 minutes, but I'll try to do my best. Basically, this is briefly the agenda that I'm going to walk through. As Horst mentioned, there are 6 different methodologies here, and I'm sure most of you in this room are not familiar with a lot of them. So I'm going to give you like a single slide overview. What would take probably a semester if you went to a university, you're going to get it in the next 3 minutes or so. And then I introduce the research protocol, briefly talk to you about what measures we've analyzed so far, give you a first pass at some comparisons between the various methods and the measures that we have, and then I'll hand over to Russ, who will go into the more predictive aspect of trying to predict which of these measures do a best job of looking at sales. All right. So the first one is the easy one. Everybody in this room must most likely be familiar with the traditional ad testing methods. And so we did basically the same kind of traditional methods in the study, which included things like measuring attitude towards brands before and after they watch an ad, getting some kind of a change measure. So these included things like familiarity, purchase intent, liking, recall and recognition, and a bunch of other measures. We also got some measures which are primarily ad diagnostic measures, with questions like asking a participant, like how relevant was this ad for the product that was advertised, or how informative was this ad, and so on and so forth. So that's the traditional part of the study. The second thing that we did was implicit measures. So some of you may be familiar with some of these measures. So these include basically trying to get a sense of what people's attitude towards the ads were, but in an indirect manner. Rather than directly asking them, what did you think about this, what we do is we use things like response times, latency measures, and so on to try and get at the same kind of preferences. So that's what the best known one is the implicit attitude test. We use a slightly modified version of this, which was suited more for the kind of protocol that we had in the study. Moving on to the other measures, again, the one that may not be that unfamiliar for most people in the room is eye tracking. This is a method that's basically used to try and get some kind of an idea about where on the screen are people focusing their attention on. So it's primarily a measure of attention and visual search. But what's also interesting with eye tracking is you can use the pupil size as they are viewing these stimuli as an index of how much arousal they are feeling. So the more aroused you are when you're watching some dynamic stimuli, the more likely that you're going to have greater dilation of the pupil. So that's another measure that we are going to use in this particular study. Moving on to the biometrics, there were two broad categories of biometric variables that we tested. One is basically EKG, which is usually a method used to get a sense of heart rate and breathing. So as some of you might know, your heart rate goes up or down depending on how engaged you are in the stimulus that's being presented to you. So we primarily use measures like heart rate acceleration or heart rate deceleration to try and get a sense of how much attention or arousal there is in the stimuli that's being presented. And the second measure is the galvanic skin response. So this is, again, used to get a sense of arousal. What it does is it measures changes in electrical properties in the skin. So the more you're engaged or the more aroused you are, you tend to sweat more, and that gets basically measured when you're using the skin conductance response. As you can see from the little graph below, it's a much more slower response, so you have to use some algorithms to tease out and be able to attribute what exactly in the stimulus is driving this kind of a response. Then we move on to the neuro measures. So we use two broad neuro measures in the study, EEG and FMRI. So EEG basically is measuring electrical brain activity on the scalp that's indirectly related to the underlying neuronal activity, right? So basically what you're trying to do is you're getting all these signals, like those little electrodes that you see on the little signal on the right. What it is a very high temporal resolution signal that tells you a little bit about consumer processes like attention and engagement. And another common measure that EEG also provides is this sense of approach avoidance behavior. Like basically, how much do you feel attracted towards a stimuli, or how much do you feel basically like avoiding a particular brand or particular kind of stimuli. And the last method is fMRI, the functional magnetic resonance imaging. What fMRI does is basically, in a non-invasive manner, captures blood flow related changes in the brain. So every time you perform a task, you need blood to be supplied to the brain to perform that task, and you can measure the changes in blood flow. So you can basically pinpoint where in the region or where in the brain is a particular task engaged currently. So here is my one fMRI slide, which usually people like to see in presentations like this. What we have over here is basically a three-dimensional view of the brain, as if I was standing in this particular angle, or rather this particular angle, depending on which screen you're looking at. And if you think of some kind of a middle split of the brain, you can look at some regions that are lying inside. So the point I want to make here is it's no longer restricted just to the surface, but you can also access regions that are hidden underneath. And what also is a very quick summary, a lay summary of what you can do, we can basically categorize regions to those that are involved with cognition or attention. You can think of regions that are involved with more emotional processing, and even regions that have to do with motivation or your intent to purchase. And based on looking at how much activations we have in these regions, we can say something about the ad that the subjects are watching at any given point of time. So how are we going to put all this together? So basically what we came up with is an experimental protocol that consisted of 4 broad phases. So phase 1 primarily dealt with traditional measures and implicit. Phase 2 measured eye tracking together with the biometrics, which is EKG and skin conductance responses. Phase 3 was EEG, and phase 4 is fMRI. And all these phases were conducted at the Center for Neural Decision Making in Temple University, which has the capabilities to basically run all these different kinds of analyses. All right. Just quickly going through the sample, basically the sample consisted of participants broadly drawn from the greater Philadelphia population, very good representation of age group, good distribution of gender and ethnic diversity. We had about 200 participants for the traditional measures, about 100 for the implicit, and 30 for all the other biometric measures that I talked to you about. All right, that brings us to the protocol. So what we tried to do in this study was to come up with a clearly well-defined protocol that was consistent across all the 4 phases. Okay? So pretty much all the phases had the same protocol, which included testing 37 ads, and it had and within them, there were basically 15 unique brands. All were 30-second spots, and these are only television ads. And participants basically watch each ad. At the end of the ad, they answer a series of traditional questions about the ad that they just watched. And then we go on to the next ad and so on. And of course, the ads were rotated across participants. And once they go through the entire set of 37 ads, they go through a set of other measures like recognition and recall and so on and so forth. OK. Sorry. All right. So what were some of the measures that we used? Like I said, again, summarizing very quickly, we used about 14 traditional measures. So these are measures that are often used in ad testing. We used 3 implicit measures, 7 eye tracking measures, 11 biometric measures from both EKG as well as SCR, 2 EEG measures, and 11 FMRI measures. And what we also tried to do was to take all these measures and categorize them into different constructs. For example, some of these are measuring attention. Some of these are measuring arousal. So what we tried to do was put these together in terms of these broad constructs and see what was happening with respect to each of these constructs. So what about the analysis? So what I'm going to do is briefly give you a summary of 2 broad ways in which we analyze this data for the first pass. First, what we tried to do was to try and understand something about the correlations across the different methods. So what is it that these methods are measuring, and is there any consistency at all across all these different methods? And the second thing that we tried to do was a more construct-based analysis. So what we tried to do, like I said before, is try to group these things into various constructs, and we tried to see, are all these different methods that claim to be measuring a particular construct doing the same thing? So those were the 2 broad ways in which we tried to break this data in the first pass. All right? So that brings me to the Uber correlation map. I'm going to walk you through this a little bit, but this is basically the main slide, right? So what you have here is a bunch of greens, yellows, and reds. When it's green, that means there's no correlation between what we are looking at. When it's red, that's very strong correlation. And when it's somewhere in between at orange or so, you have a moderate correlation. And the way I've broken this graph down is basically looking at traditional measures compared to eye tracking, bio, and EEG fMRI. Okay? So what's very obvious at first pass is each of the methods, the measures within them are highly correlated. So you see a nice bunch on the top, which shows that a lot of the traditional measures are correlated with each other. Then in the middle, you see a bunch of, again, red spots, which shows that most of the eye tracking measures are correlated with each other. And most of the biometrics are correlated with each other, and most of the fMRI measures are correlated with each other. But I'm sure most of you in this room are more curious about what do these methods add to what I can find out from the traditional methods that I have, right? So what I'm going to do is I'm going to break this graph down only in the top 1/3. And I'm going to try and compare what do each of these methods do in terms of what I can learn from just the traditional methods. Okay? So the first pass of this graph is just looking at traditional versus traditional. And as you can see, there is a lot of correlation between the various traditional measures that people obtain. Right? So these measures tend to be highly correlated in some cases, but there are also some other things that you learn, as you can see from the non-correlated areas. But what's interesting is once you go into other things, for example, if you compare all the different eye tracking measures that we have with the traditional measures, there is some correlation. You can see a few orange spots here and there, but there is a lot of new information that you're learning. That's the simplest way to read this graph, right? There are additional things that you stand to gain beyond what you get from just the traditional measures. Not everything we have is going to be correlated with the stream of traditional measures that we have. And the same is true with biometrics, and also the same is true with fMRI and EEG. Okay? So basically what you can see is there is a little bit more measures that are correlated, which is good. You get some kind of face validity that what the brain is doing is obviously what the behavior is, but also there is a lot more additional information that you can gain by looking at data from both fMRI as well as other kind of biometrics. Okay? And remember, the other thing we were also interested in is basically trying to see how these measures work with a construct. So what I have in this graph is rather than just looking at all the measures across all the methods, I only tried to highlight all those measures that highlight attention, right? So for example, if you're thinking about heart rate, you'd probably think the greater heart rate deceleration, the more the attention that you're paying to a stimulus, right? Or with terms of eye tracking, you can look at how much attention was being paid to a particular part in the screen. So we can take all these different measures and see what is the correlation between all these measures of attention. And again, as you can see from this graph, there seems to be some things that are correlated, but a lot of things are still different, right? So it's not that everything is really highly correlated with each other. So again, the take-home message, if I had to summarize these two slides into one, is that most biometric and neurological measures are weakly related to traditional measures. So there's some kind of a correlation, but at the same time, there's also a lot of variance that's not just explained by the traditional measures. And second, and more importantly, even different measures of constructs like attention, right? So you may think, OK, I'm measuring attention using method A, method B, method C. But what's interesting is, again, these are only weakly correlated. In other words, they do not necessarily measure the same thing, and there may be more to learn from these additional methods than what we can get by simply looking at the traditional methods, right? So at this point, I'm going to hand over to Russ, who will summarize what we can learn from these measures in terms of predicting the objective sales data.
OK, thanks very much, Vinod. The handing of the clicker is very much a metaphor for the way the project worked in a lot of ways, where, as you saw, the Temple team developed some very fascinating results from their lab work. The role of the NYU Stern team was to take the results that they found and to try to get some insight into how these measures affected traditional measure of sales response to advertising, which is an elasticity. And I will go through our multi-stage, actually two-stage approach to doing this as we use their data as a way of trying to answer some of the questions that the sponsors wanted to address. About a year ago, we had a meeting here at the ARF with sponsors and potential sponsors, and they were interested in the kinds of things that Vinod showed you, but they're also interested in whether or not different types of neural measures actually did a better job explaining ad effectiveness than maybe more traditional measures, which most of you have been using for many years. So that was really the goal of this second part of the study. And it's a two-stage process, as you can see here. The first stage took data from 5 of the sponsors that gave us both ad executions and data on market share, GRPs by week, and other good data that we would use to estimate what has been used in this conference to be called a marketing mix model. So we estimated kind of a traditional marketing mix model, although we had a couple of nice features that were mentioned in the sequence presentation this morning. We had by execution, and we had weekly GRPs for those executions. The problem is some of the executions ran for only a short period of time, so sometimes we had to merge or aggregate across execution that had the same theme. But we estimated basically by sponsor a marketing mix model with market share as the dependent variable, GRPs for the tested executions as independent variables, and depending on the client, we had some other data, price, GRPs for other executions that they ran, and a number of other independent variables as well. So what we called the stage 1 estimates, we looked at TV advertising elasticity, as you can see, by creative execution, trying to control for other factors that we could. And interestingly, we found a fair number of the elasticities were significant and positive. Those of you who do statistical work in this area know that if you could get 100% of the elasticities significant, that would be pretty amazing. Some of them were not, some were 0. A good share of them were significant and positive. And interestingly, they were in the range of the elasticities that I've seen over a number of years of looking at TV advertising elasticities. There have been a number of published studies in the Journal of Advertising Research and other journals that have done what are called meta-analyses across large numbers of advertising studies, behavior scan studies, and other type. and I found elasticities in the 0.1, 0.15 range. Our elasticities were approximately in that range, with some higher and some lower. So it was a nice kind of check that we were getting reasonable elasticity that had been found in many other studies. So think of the following. What we have from the first stage for 5 of the sponsors, we have estimates of the elasticities of approximately 5 executions per sponsor. Now, one of the sponsors we could not use in the second stage because what we predicted there was not market share sales. It was an unusual dependent variable. We did not use it in the second stage. So think of like a dependent variable being an advertising elasticity. We had 21 advertising elasticities. And the second stage in this part was very simple. It was 21 observations as a function of the neural measures that Vinod just described, plus traditional measures. And again, the idea was to find out, whether or not these things significantly predicted ad elasticities, because remember, we're taking these measures at the individual level, right? Somebody's in an MRI tube, looking at an ad, does what they see aggregated over the respondents in that sample have anything to do with market level, in fact, US level, advertising elasticities. So the second stage then had the elasticities as the dependent variable, independent variables, the neural measures plus traditional. Only 21 observations, so as Vinod said, we had to cut down the number of variables significantly, and still, as you can see, we were able to have a few degrees of freedom in the second stage. So the question is, what did we find? And of course, that is the sort of the bottom line here, as they say. What we found was that the traditional measures explain the most variation in the advertising elasticities, followed by FMRI. And in addition, biometric measures sort of came in third. And the good news is that there seems to be some additional variation in advertising elasticities or sales response to advertising based upon these other measures beyond what you've been doing for a long time. All right, now, we're not concluding today that other methods aren't useful. We've only really started the analysis. And as you can see, we need more analyses to assess the implicit measures and biometric and EEG measures in addition. We haven't concluded all of our analysis. All right, so in summary then, it's really the first study to link the kinds of measures that the Temple team did to actual marketplace behavior. There have been plenty of other studies, as you know, that have taken a look in a pretest copy situation, how different parts of the brain light up to different kinds of executions and skin response, and some of those have been around for a long time also, as you know. But what's really good to know is that beyond the traditional measures, all right, which hopefully do work well, and as you can see here they do, neuromarketing methods such as FMRI also predict sales. And so, we're really at the beginning of this. This is kind of like a framework for trying to assess whether or not these additional kinds of lab measures actually do add value over traditional measures. And as you can see, adding the neurological measures actually improved the predictive power of the model. So there's a lot more to come, as I have on here. This is a framework, and thank you very much for your attention. Okay?